Hello everyone. In this segment, I'll discuss about isolation and characterization of phytoconstituents. Let's forward to our first slide. This talks about the general introduction of the current topic. Phytoconstituents are the bioactive chemical compounds found naturally in plants. These include alkaloids, flavonoids, terpenoids, glycosides, tannins, saponins, steroids, and phenolic compounds. Isolating and characterizing these constituents is very much essential for drug discovery and development, which ensures the development of the newer molecule, standardization of herbal products, which is very much needed for the safety security of the herbal product specifically that we are going to use, understanding of therapeutic actions and mechanisms that is related to the pharmacological and therapeutical applications and aspects, as well as the uh, assurance of the product related to their activity study and more specific function towards human civilization. So whatever product kind of chemical compounds I have mentioned, like alkaloids, flavonoids, terpenoids, glycosides, these all are considered as the secondary metabolites. And if I talk about the phytoconstituents, then isolation of the phytoconstituents is a very important uh, kind of character which actually plays an immense role in the development of newer molecules, in formulations, in the preparation of formulations, and to establish a patent also. There are several steps involved in the isolation and characterization of such products, which divided into different segments. Like, first starts with the collection and authentication of the plant material. It basically speaks about how do you collect the natural products from different regions based on their geographical distributions, based on the seasons, based on their uh, full blossom condition. Uh, Whenever we are looking for a particular part of the plant or specific part of the plant, what will be the best time to collect all those products? So this is all about collections. And regarding collections, another thing is very much important. After collecting such product, how do you store the product? Means storage of the material, plant material or natural products. It's equally important, because until and unless after just collection you are not able to store the product properly, degradation may start. Degradation which leads to the inferiority of the product or substandard of the product. Authentication is something which is scientifically very much sounded term, and it is uh, needed also at the same point of time whenever you are going to establish any of the plant material or animal product, whatever kind of natural products, in fact, from different origin, of course. You have to authenticate the product properly, whether you have collect. If suppose, means if you are claiming that I have uh, isolated or extracted such products from tulsi leaf, I have prepared this formulation by using vasaka leaf. At the same point of time, you have to prove uh, that whatever you are claiming that your product is from, by using raw material as tulsi leaf or vasaka leaf, you have to establish that one by means of a valid documentation. That is authentication is all about the plant material and animal product too. So in case of plant material, uh, if you want authenticate the product, the best way in India to authenticate the product is to prepare a herbarium according to the instruction given by the Botanical Survey of India, there are several branches of Botanical Survey of India. They will help you to authenticate the product. Just forward your sample to nearest Botanical Survey of India office. They will investigate your product. They will, after that, they will provide you a certificate in terms of where all the taxonomical classifications, all the taxonomical parameters, features related to the product will be written over there. You will get the entire concept about the proper authentication certificate of the plant material. Then moving towards extraction. Extraction is something where we are collecting the plant samples. After collection, after proper processing, it is subjected to be the extraction if it is needed, of course, by using a suitable solvent. The factors affecting extraction, most important thing, that the coordination between solvent and the drug. Uh, you have to choose the solvent according to the polarity basis, as well as the nature of the compound you are going to be extracted, along with the nature of the solvent that you are choosing. So this is all about extraction. Then fractionation and isolation, where we are specifically targeting for using uh, just one specific or different specific solvents according to their specifications, and targeted isolation process or targeted product to isolate such product and just establish their efficacy. Purification is considered as one of the important feature as far as the entire process is concerned, because until and unless you are not purifying the product properly, your product may be maybe in such position where it stands along with the different impurities. It affects a lot in case of animal studies, furthermore in case of phytochemical screening, or in case of different analytical process. uh, so purification is very much important. Characterization is something which is related to along with the proper physicochemical character, chemical character, biological character of the compound, which ensures the product more specifically where it actually belongs to. The biological evolution, which is very much important, because until and unless we are not biologically evaluate the product, we cannot assure about the activity or the efficacy of the product for a for procurement of a single disease or number of diseases. So we have to choose specific animal model to carry on our study. We have to go for in vitro and in vivo study. Then after those study, toxicity determination is very much important. How much it is toxic enough to our life, to our body. So after clearing all those parameters, we can go for the clinical trial. Then collection and authentication of plant material in detail. What I can say, that's correct. The botanical identification is crucial. Herbarium preparation and comparison with authenticated specimens, drying, grinding, and storing under appropriate conditions as per the procedures given. Isolation techniques. In isolation technique, it is related with the phytochemical screening method, where uh, qualitative test for major groups like alkaloids, tannins, then glycosides, these are, or any of the secondary metabolites. We have to perform all those tests. uh, for example, for alkaloid containing drug, we must perform Dragon-Draft test, Mayer test, Haggard test, Wagner's test. So that will confirm about the presence of alkaloid in the given product. If it is yes, the test definitely go in with a positive manner. If it is if alkaloid is not present over there in the given sample, then it will give negative result. Then moving towards chromatographical techniques, which are used to separate individual constituent present in a mixture. As far as the extracted material is concerned, in those cases we are assuming a product which is little concentrated, and where in the extracted mass different components which can be easily extracted out from the plant material, given plant material, it is mixed together and present as a mass. So this, from this kind of mass or mixture, whenever we are going to isolate the product or individually characterizing the product, the first preliminary technique which we can apply, that is chromatographical separation technique. Different types of chromatographical techniques are there. Among them, some important techniques are mentioned below. Starts with the column chromatography. It is based on the polarity and adsorption therapy, means surface phenomena adsorption, that starts AD. It is not AB. In case of absorption, it's a systemic phenomena, whereas adsorption is kind of surface phenomena. So commonly, stationary phases like silica gel G, which is made up of silica gel G or alumina. Mobile phases which can move. In case of stationary phase, it is fixed. In case of mobile phase, which can move. So solvent system must be there, and it is based on the polarity. It must be it must match the coordinates. It must maintain the coordination along with the extracted product and the used solvent. Then thin layer chromatography, which is commonly or popularly known as TLC, which is a quick and inexpensive process, of course, and used for monitoring fractions. Then in case of preparative TLC, which is performed frequently in the practice session of different labs, where it is used for isolating small quantities. uh, then we'll move towards HPLC, which is known as high performance liquid chromatography. It is precise and efficient, of course. uh, Both analytical and preparative works we can perform the HPTLC. In case of gas chromatography, which is known as GC, it is useful for volatile constituents. And in case of Coulter current chromatography, there are no solid phases. It is very much useful for large scale separations. Mm, uh, as far as the purification process is concerned, the most popular purification process we can adopt, like recrystallization, then repeated chromatographical process, then dialysis for higher molecular weight compounds, and solvent partitioning, means it's a liquid composition kind of purification process. uh, if I talk about the characterization of different phytoconstituents, I will say it may contain the physical characters. We can go along with the different physical characters, which includes color of the product, odor means smell of the product. Melting point is one of the vital uh, point to be considered. Melting point, because it indicates the sharp and pinpoint temperature where the product starts to melt, and it indicates the actual purity of the compound also. So melting point, it's a very means helpful parameter in such cases. Solubility, which is again related to the polarity, and solubility which is also related to the chemical character, affinity of the solvent or product. Those are going to mix with each other, or uh, nature of the solute and solvent both. So it all works together in solubility. Whatever we are choosing, doesn't matter, but these parameters are considered as one of the vital parameters. uh, then moving towards some advanced techniques like spectroscopy techniques, which is structured by UV visible spectroscopy or UV ultraviolet visible spectroscopy, where different wavelengths has been used, long wavelength, short wavelength, and on the basis of the specific wavelength, we have to judge the intensity of the product, and such the purification of the product can be judged along with the standard, where it gives information on conjugated systems. Moving towards the next spectroscopical analysis method, which is known as infrared or IR spectroscopical method. It is very much helpful for identifying the functional groups. Functional group present in the given material. It may be present in the extracted material, extracted compound. It may be present in the isolated compound. It may be present in the from some obtained mass. So by using such product on the specification of the instruments, we can put the sample over there and we can identify the functional group as well. Next, moving towards nuclear magnetic resonance or NMR spectroscopy, which is very much helpful for 1H NMR and 13 carbon NMR for structural elucidation process, where specific structures for hydrocarbon can be elucidated very in a very positive manner, and it is very much helpful for such hydrocarbon containing products as well. Specifically, those are obtained from different plant materials. It becomes mandatory in such cases to identify the product by using NMR spectroscopy. 2D NMR for complex structures. It is very much useful for today, where the structure become little complex in nature, to identify their functional groups and their nature of bonding, like hydrocarbon bonding or any other bonding. So 2D NMR for complex structure is very much useful and helpful by the use of NMR spectroscopical method. Then mass spectroscopy, where it helps to determine the particular molecular weight and fragmentation pattern of the given sample. It is again applicable for extracted or isolated material, but more specifically in case of isolated material, if purification has been done in such cases, it gives very accurate value. So it helpful for the determination of molecular weight and fragmentation pattern. In case of X-ray crystallography, it is for 3D structure or crystalline compounds. Specifically and more specifically, uh, it is a very much unique method in for crystalline compounds, importantly, and in such process of such methodologies are helpful in today's era, where there is a big doubt or question or purification process becomes stuck along with the crystalline compounds. So chromatographical fingerprinting method, if I talk about this, then HPTLC or HPLC profiles for standardization is need to be considered enough for chromatographic fingerprinting method. Um, then moving towards the bioassays and activity screening, which is optional also, but until and unless we are not performing such processes, we cannot establish a thought related to a specific biological evaluation process. When we are talking about biological evaluation process, it always refers to something that is related to the targeted activity study, and which will furthermore carry forward towards preclinical trial, clinical trial, and furthermore. So, in such cases, most popularly antioxidant study, antimicrobial study, anti-cancer study, or anti-inflammatory activity, anti-diabetic activity, anti-cancer activity, means there are several anthelmintic activity. So number of activities are present over there. So these are very much helpful to identify the kind of product that we are going to use by getting that extracted material or isolated, and just followed by carry on such processes. Different challenges in phytoconstituent isolation processes are there. First of all, the complexity of the plant matrices, that that is very much important to discuss, because plants itself is known as the most complex biochemical laboratory. So whenever we are going to extract or isolate such product, uh, it's quite natural that maybe possible in maximum cases we may face some complexity of the plant matrices, which is of greater thought, means until and unless we are not simplify the structure properly, you cannot establish the actual elucidated structure of the given compound. Then low concentration of active constituent, that is also important. Degradation during processing, which may lead to the lowered potential level of the drug. And then reproducibility issues, which may affect also furthermore. Applications: the newer drug discovery, quality control of the herbal medicines or natural products, nutraceutical development, understanding pharmacological mechanisms. These are the applications which is considered as the most important one. And last, the conclusion, where I would say the isolation and characterization of phytoconstituents is a foundational aspect of natural product research. It combines classical and advanced techniques to explore and utilize the medicinal potential of the plant material or any of the natural products as well. A thorough understanding of these techniques enhances the development of safe, effective, and standardized herbal therapeutics. Thank you very much.